All right, let's go ahead and get started on chapter 8. This is probably going to be the most taxing of all of the chapters in the book. So I want to give you um, a really quick reading assignment before you start watching this PowerPoint. I'd like you to read sections 8.1 through 8.3 in the textbook. You're probably not going to understand a good portion of it, but it will give you kind of a roadmap of the direction that we're going. Also, on a number of the pages, there are these purple boxes that say Insight. I'd like you to read Insight 8.1, 8.3, and 8.4. These will help you understand some of the different parts of metabolism and give you a little bit deeper clue about what's going on. So if you haven't read this chapter, please take some time to read it before you start watching this PowerPoint. And then once you've read it and you watch it, you'll see, you'll have, I think, a better understanding of what's going on. So for those of you that have read it, let's go ahead and move on. For those of you that haven't, please pause, read these, and come back and watch the PowerPoint when you're done. Metabolism is essentially all of the chemical reactions that take place in the body. Any kind of physical work that goes on in the body has to have some kind of energy source, and that energy source is going to come from a metabolic process. We can break metabolism up into anabolic reactions and catabolic reactions. Anabolic reactions are going to be those reactions that build things. They are going to be biosynthesis reactions where they are going to take smaller molecules and build larger structures out of it. These are building reactions that are going to require energy. One of the ways that you can remember at what an anabolic reaction is, think about anabolic steroids. If you have someone who takes anabolic steroids, their muscles get bigger and bigger and bigger. They're building muscle. So anabolic reactions are building reactions, and they are going to require energy. That energy has to come from some place, and that place is through a catabolic reaction or through catabolism. These are reactions that are going to break bonds of larger molecules and produce smaller precursor metabolites. Catabolic reactions, when you're breaking bonds, you're releasing energy. So the energy of anabolic reactions will generally come from a catabolic reaction. So these catabolic reactions will release energy. There are several functions of metabolism. The first is that they are going to assemble smaller molecules into larger molecules. We're going to have those anabolic reactions that are going to produce cell structures. They're going to produce macromolecules, clear up even to a single-celled organism or a single cell. These reactions will also degrade macromolecules into our smaller molecules, and they will then yield energy. And that energy can be used for any kind of other physical work that needs to be done in the cell. Energy in metabolism can be conserved as well in the form of ATP or in the form of heat. You're going to see that the pathways we're going to talk about as far as metabolic pathways are concerned are the pathways that are going to help us generate ATP. Here is a graph that's in your book. It is actually, pardon me, it is actually on page 200, and you can see from one catabolic reaction of glucose, where we're going to take glucose, we're going to break it down. We are going to get three major components out of it. We're going to get precursor metabolites or precursor molecules, these molecules that we can use to build building blocks or macromolecules. We're going to get ATP, and we're going to get something called reducing power, or NADH, and this is a coenzyme that is going to help us to produce a significant amount of ATP. From these precursor metabolites, now we can go through a series of anabolic reactions that will help us build up clear through our cellular organism. So these anabolic reactions will feed off of the ATP that is produced through this single catabolic reaction. A major component in metabolism is the enzyme. Metabolism relies on enzymes for the proper functioning of metabolism. If you're looking at the pictures in your book as we go through glycolysis and the Krebs cycle and all of those, you're going to see that there are steps. These are pathways, and each one of the steps in these pathways is catalyzed by an enzyme. Enzymes are catalysts, meaning that they are going to increase the rate of chemical reaction by lowering the amount of energy that reaction requires. They work without becoming a part of the products of that chemical reaction. So they can basically bind to a reactant, help reduce the energy required for that reactant, produce a product, and then go back and bind to a new set of reactants and keep the process going as long as there is a substrate available to be used, or excuse me, a reactant that's available to be used. So these things are going to increase the rate of a chemical reaction. Every chemical reaction has an energy of activation, and this energy of activation is the amount of energy required for a chemical reaction to proceed spontaneously. So this energy of activation is the amount of energy that has to be overcome before this reaction can take place. We can achieve the energy of activation three ways. We can add heat, which will increase the thermal energy and the molecular velocity of the reactants in the chemical reaction, which will in turn break bonds and cause products to be produced. We can increase the concentration of reactants to increase the rate of molecular collisions. When these molecules bind or collide with each other, they will break bonds and chemical reactions will take place. Or we can add a catalyst like an enzyme and lower the amount of energy required for that chemical reaction to take place. An enzyme is going to promote a reaction by serving as a place or a physical site where a reactant molecule can be positioned for a particular interaction with other molecules. When we talk about reactants and enzymes, those reactants are something called a substrate. We'll talk about what a substrate is here in just a little bit. As far as enzyme structures are concerned, most of them are protein. They can be classified as a simple protein, meaning that they are an enzyme that is made, or excuse me, they can be a simple enzyme, which means that it is a protein only. There is no other molecule attached to it. This is just a simple protein by itself. Conjugate enzymes contain protein, but they also are going to contain some sort of non-protein molecule that attaches to the enzyme, to the protein portion of the enzyme. These conjugate enzymes are called holoenzymes, and these holoenzymes have two parts. They have the protein part, and then they have the cofactor or coenzyme part. The protein portion of a holoenzyme is called an apoenzyme. Okay, so when you see in your book that it's talking about apoenzymes, you know you're talking about a conjugate enzyme where you have an apoenzyme, the protein portion of that conjugate enzyme. For these holoenzymes, you're going to have one or more cofactors or coenzymes bound to this protein. A cofactor is going to be some sort of inorganic element like a metal ion that is going to help in the function of the enzyme. A coenzyme is an organic molecule that is going to act as a carrier molecule that just kind of rides along that chemical reaction with the enzyme. Here's a picture of what coenzymes and cofactors and apoenzymes. If you look here at our first picture here, we have the protein portion, which is our apoenzyme, and it is attached to some sort of metallic cofactor. Here the apoenzyme is attached only to a coenzyme, and here we have the apoenzyme attached to a coenzyme as well as a cofactor. All three of these kinds of configurations are legitimate conjugate enzymes. One of the characteristics about an apoenzyme is they have specificity. They are proteins, and because they are proteins, they exhibit those same levels of molecular complexity that a protein does. They have a primary, a secondary, a tertiary, and a quaternary organizational level that will allow them to each have its own specificity, its own active site. The actual site that a substrate binds to, or that the reactants of a chemical reaction bind to, is a groove in that enzyme called an active site. Your book also calls it a catalytic site. I find active site kind of gives you a better idea of what exactly goes on in that portion of the protein. It activates the chemical reaction. So you will hear me calling it an active site rather than a catalytic site. I used the term substrate a little bit ago. Substrates and reactants in a chemical reaction are the same thing. A substrate is the specific molecule on which an enzyme acts. So you have a substrate or a set of reactants that binds to the enzyme, and that enzyme then will initiate a chemical reaction, and you will end up then with a product. Here we have our picture of our structural levels of protein. Remember, we have our primary, then our secondary, our tertiary, and our quaternary levels. If you look here, you'll see on the primary and secondary level, you do not have any active sites on those primary and secondary levels, because remember, those are not functional proteins. Once we get to the tertiary level of the protein, we're going to start having some of these grooves and crevices in the protein where active sites will be created for substrates to bind. Obviously, in a quaternary protein, you're going to have a number of active sites, and you can then have that enzyme binding to a number of different molecules at the same time. All of those molecules will be the substrate of that enzyme. For a chemical reaction to take place with an enzyme, you're going to have the enzyme and the substrate binding together. And once they do, you will have the enzyme binding to the substrate at the active site of the enzyme. Once that enzyme and that substrate bind together, you have something called the enzyme substrate complex. When that is created, now you have the molecule that is bonded together that will allow for the energy of activation to decrease, and you end up at the end with your final product. One of the ways that you can think about the enzyme and the substrate is a lock and key fit. Each enzyme has a single or a very limited number of substrates, and each enzyme will bond only to those substrates. So it's not going to be bonding willy-nilly to any molecule that's out there. I have a key to my house, and you have a key to your house, and my key will probably slide into the lock at the front door of your house. But is that lock going to turn? Is it going to open? No, because my key is not specific to your lock. But if I go put my key into the lock on my front door, it turns easily and I can open the door. The enzyme and the substrate act the same way. The enzyme is the lock, the substrate is the key, and that key has to fit exactly or relatively close that it will cause that chemical reaction to take place. When you have enzymes and substrates bonding, the bonds are very weak and easily reversible. So when you have that enzyme substrate complex made, once the products are produced, it can be released easily, and you don't have to spend a lot of energy breaking bonds holding those products to the enzyme. You have a number of different molecules that are going to support the functioning of enzymes, and one of those are those cofactors that we talked about, those metallic cofactors. You see here we have a number of them: iron, copper, magnesium, manganese, zinc, cobalt, and selenium. These are going to activate enzymes, and they are going to help bring the substrate close to the active site so they can bond together, and then they will participate directly in the chemical reaction with the enzyme and the substrate. It's going to help with the whole production of the product at the end of the chemical reaction. You also have coenzymes, and these coenzymes are going to be organic carriers, and basically they are going to work with the enzyme to perform any kind of alteration on the substrate that will help it to eventually produce the products that are going to be at the end of the chemical reaction. Some of these coenzymes will remove things like functional groups, or they will remove hydrogens or electrons, and they will carry them from one molecule to another. So they act kind of like a molecular basket where they will remove one set of molecules from one set of reactants and take them to another chemical reaction where they will be deposited into that chemical reaction, and we will do anything that needs to be done then once they get there. So these coenzymes are molecular carriers. They're going to act like baskets from one chemical reaction to another. A number of our vitamins are very important coenzymes, and most of these we are going to get, and the cell is going to get from its environment. We can classify enzymes based on their function. We can do it by site of action, we can do it by type of action, or we can classify by substrate. If I have an enzyme that is in a class called the lipase enzymes, this would be a substrate type of an enzyme, and the substrate would be a lipid. If I have a transferase enzyme or transferase class of enzymes, this would be type of action of an enzyme, meaning that I am using enzymes to transfer molecules from one place to another. Site of action, these are going to be our endo and exo enzymes. These are going to be enzymes that work either inside or outside of the cell. So let's look at some of the ways that we can look at endo and exo enzyme action. Essentially, you're going to have these enzymes working either inside or outside of the cell. Exoenzymes are going to break down molecules outside of the cell, so those molecules can be taken then, those products can be taken into the cell and used inside the cell. Endoenzymes are going to break down molecules inside the cell, and the products can either be used inside the cell or they can be taken outside of the cell. Exo means to exit or to leave, so these are going to be on the outside. Endo means to enter or inside, so those are going to be ones that are on the inside of the cell. Enzymes have a rate of reaction, and not all of them produce equal amounts of product at equal rates. You have enzymes that are what we call constitutive enzymes, and these constitutive enzymes are always present in a relatively consistent amount. These enzymes are never turned off. They are never regulated. But we do have some enzymes that are regulated because we only need their products at certain intervals. And so we're going to have to either induce or repress the response to these enzymes. We don't want to have a product that we can't use and it gets wasted, as well as the energy being wasted to produce those enzymes. So we either have to induce or repress the action of those enzymes in response to the concentration of a substrate. The cell is only going to produce these enzymes when they're needed. So if we look here, you can see with figure A here that constitutive enzymes are made at a constant rate. It doesn't matter if we add more substrate or if we take away more substrate. These enzymes are going to be made at a constant rate. Some of the constitutive enzymes that you have in the cell are those constitutive enzymes that are going to regulate like metabolism and the cells that take us through glycolysis, the Krebs cycle, and the electron transport chain. All of those are going to be required all of the time. So those are never going to be turned off or turned on. If you look over here at figure B, you can see that if we have regulated enzymes in the presence of more substrate, we can induce enzyme action, and those enzymes then will work until the substrate is removed, and once those substrates are removed, then the enzyme is going to be repressed. One of the reactions that we need to understand that enzymes do, and this is basically in all of metabolism, are these reactions that we call oxidation reduction reactions. These are also known as redox reactions. Basically, in a oxidation reduction reaction, a compound is going to lose an electron, and that compound then is going to be oxidized. The compound that receives the electron then is going to be reduced. You must have, if you're going to have an oxidation reaction, you must have a reduction reaction. You can't have one without the other. These are very common in the cell. Most of metabolism is a series of oxidation reduction reactions. So it's very common. Some of the important components are the enzymes, oxidoreductase enzymes. These are going to be enzymes that are going to run the whole oxidation reduction reactions. All of the oxidation reduction reactions that have enzymes attached to them are going to be these oxidoreductase enzymes. If you look here at this picture, pardon me, you'll see that we have sodium and chlorine. This is a very common oxidation reduction reaction. Sodium has that outside valence electron, and it only has one. It would love to have a full valence shell, and the easiest way to do that is to give up its electron. So once it gives up its electron, it is in its oxidized state, and it gives it a plus charge. If we look at the chlorine that has 7 electrons in its outer shell, and it would love to have a full shell, then it can accept that electron from sodium, and now it has a full 8 electrons in its outer shell. But what happens here is that we are reducing the charge. We're going from a 0 charge to a -1 charge. So this is why we say that it is a reduced molecule or a reduced atom. So oxidation is going to give away an electron. Reduced atoms are going to take on that electron. And the reduction is basically because of the reduced charge. Okay? In metabolism, we're going to use coenzymes NAD and FAD to be our reduced coenzymes. You have a number of different microbial enzymes in disease. A lot of pathogens are going to secrete a number of exoenzymes, and those exoenzymes will bind with tissues, cause tissue damage, and you end up with symptomology. Some of these enzymes can help them avoid host defenses and promote multiplication in specific tissues so that you end up with increased numbers of bacteria, and then you begin to have what we call supraclinical numbers, or the number of bacteria that are required to begin to cause symptoms. These exoenzymes can often be called virulence factors or toxins. Some examples of the bacteria that produce exoenzymes: Streptococcus pneumoniae. This is going to cause Streptococcus pyogenes. This causes skin and throat infections. This is the bacteria that causes strep throat. Basically, what it does is it produces an enzyme, an exoenzyme called streptokinase, that will digest blood clots, and it will help then invade the tissues in the back of the throat, and it makes the throat very red and sore and bloody. Pseudomonas aeruginosa. This also causes respiratory and skin infections, and it produces a couple of enzymes called elastase and collagenase. And what these do is they basically destroy the proteins elastin and collagen that are part of the connective tissue, and this can lead then to lung and burn infections. When you have patients with severe burns, Pseudomonas aeruginosa is one of those that gets in there and begins to kind of chew away at that tissue. Then finally, you have one, Clostridium perfringens. This is going to cause something called gas gangrene, and essentially what it does is it produces lethathinase C and lipase, and this is going to cause damage to the fatty tissues in the membranes, and it's going to cause the cell then to die, which is basically what happens is the tissues become necrotic, and then you have to end up cutting off the limb or the tissue or whatever has become gangrenous. Enzymes are protein, and because they are protein, they are going to be influenced by the environmental factors that influence the bacteria. They are generally going to operate under the natural temperature, pH, and oxygen pressures that of the organism's habitat. But when you change those normal conditions, they become unstable. And because they are proteins, they are subject to the same environmental factors, so of instability. Once you have that unstable state of that enzyme, you have something called denaturation. And essentially what happens is you have bonds that hold the proteins together begin to break. They lose their original shape of that apoenzyme. That shape is broken, and if it loses its shape, it loses its function. So temperature, pH, oxygen, those can all cause some changes in the function of an enzyme. We need to be able to regulate our enzymes. Those enzymes have to be turned on and turned off when we need them. Most metabolic reactions have a number of multi-series steps in a pathway. Each step has got a specific enzyme that's going to catalyze that particular step. Every pathway has at least one enzyme within that pathway that's going to act as a pacemaker. It's essentially going to keep the cell from running too fast or too slow and allow a consistent regulation of those enzymes. It's going to set the rate of that pathway's progression. If it needs to be turned off, it's the pacemaker enzyme that's going to turn it off. If it needs to be turned on, it's going to be the pacemaker enzyme that turns it all on. So you have these enzymes within these pathways that are going to keep those pathways running the way they should. If you look at the enzymatic pathways, there are 4 different routes that these can take. They can be linear, where you have A to B to C to D and so on, or they can be cyclic, like the Krebs cycle. The Krebs cycle is a cyclic reaction. You start with a particular molecule and you end in the same place. You may have products that leave the cycle. You may have products enter the cycle, but it's still very cyclic. You have divergent pathways where you start with a single linear pathway and at some point they divide and you end up then with 2 pathways. Glycolysis is a divergent metabolic pathway. Then you have convergent, where you have 2 pathways coming together at a single point, and then it becomes a single linear pathway. I said earlier that we needed to be able to control our enzymes and that enzyme action, and there are a couple of ways that we can do this. We have something called competitive inhibition, and this is where the cell supplies some sort of a molecule that is going to be the same shape or same chemical makeup as the normal enzyme substrate. When the competitive inhibitor binds to the active site, it blocks the ability of the substrate to bind and the enzyme is turned off and no reaction proceeds. If the normal substrate binds to the enzyme, then the enzyme begins to activate, that reaction takes place, and we end up with product. So if product needs to be turned off, then that competitive inhibitor is going to bind to the active site. It's going to block the substrate from binding, and the reaction is going to be blocked and no product will be made. Non-competitive inhibition is much like competitive inhibition, where we're going to stop the production of a product, but it happens in a totally different way. Many enzymes have 2 binding sites. One is the active site and one is the regulatory site. That regulatory site will be an area where you will have a molecule, a regulator molecule, bind to that regulatory site, and when it does that, it causes a conformational change in the shape of the active site. And if that shape of that active site changes, now the substrate can't bind to the active site and the pathway or the enzyme is turned off. Sometimes the end product of the reaction will be the regulatory molecule that binds to the regulatory site. But if you have a regulatory protein or a regulatory molecule bind to that regulatory site, the active site changes shape and everything gets turned off. There's one type of regulation that your book doesn't really spend a lot of time talking about, but I do think it's important to talk about it, and that is something called feedback inhibition. And essentially, feedback inhibition is going to help the cell not waste chemical resources. Basically, what happens is you have a chemical pathway where the end product will feed back onto the first enzyme of that pathway and turn the whole pathway off. If you look here on the diagram on the side here, we have a substrate that binds to a particular enzyme. So when the substrate binds to the enzyme, we have substrate enzyme complex number 1. That makes intermediate molecule, intermediate product A. That intermediate product now becomes the substrate for enzyme 2. I've lost my cursor here. So the product that was made from the first reaction now becomes the substrate for the second reaction, and we're going to make product B. Product B now becomes the substrate for the enzyme of the third reaction, and we get our end product. Once we've accumulated enough of this end product, it's going to come back and feed onto the allosteric or the regulatory site of the original enzyme of the pathway, the first enzyme of the pathway. When that binds to that regulatory site, it causes the change in the shape of the active site. The substrate can't bind and the pathway shuts completely down. So feedback inhibition, because it binds to that regulatory site, is a form of non-competitive inhibition. It's not competitive because if it were competitive, then we would have our product be the same as the substrate at the beginning of the pathway. So it feeds back onto that regulatory site and shuts the whole thing down. We have a number of different ways that we can gain our energy. We have two different types of reactions. We have exergonic reactions and we have endergonic reactions. Those exergonic reactions are going to be reactions that will release energy as they proceed. These are going to be reactions that produce heat or produce energy. These would be all catabolic reactions. Then you have endergonic reactions. These are reactions that are driven forward with the addition of energy. These are going to consume heat, and these would be our anabolic reactions or our biosynthetic reactions. I'm not really fond of this picture, and I probably will take some time to draw a new picture for you. But basically what this is showing us is that when we begin metabolism with our molecule of glucose as our starting fuel, we are going to go through a series of oxidation reactions, a series of enzyme catalyzed reactions, and we are going to release electrons and produce energy all the way through this series of chemical reactions, and we're going to end up with end products of carbon dioxide and water that are at a very low energy. So we start out with a molecule with a high energy, meaning that we have a lot of free energy, particularly in the form of hydrogen, and we're going to drop down here and we are going to be down to these very low energy molecules of carbon dioxide and water. So in aerobic respiration, our end products at the very end of the pathway are going to be carbon dioxide and water, and these are going to be at extremely low energy. We talked a little bit about coenzymes as carriers, molecular carriers, but we have these molecular shuttles, these electron carriers, that are going to be repeatedly oxidized and reduced. They're going to accept and release electrons, and sometimes even in the form of hydrogen, through a whole series of processes. These electron carriers, are going to facilitate a lot of the transfer of energy in the form of oxidation reduction reactions. They're going to be passing electrons down through a series of carriers and produce energy as they go. Most of these carriers are going to be coenzymes that are going to transfer both electrons and hydrogen throughout these metabolic pathways. The most common coenzyme carrier is the coenzyme NAD+. We'll talk about this a lot here as we move on through this PowerPoint. Another product within the pathway, and this is probably the most important product that we're trying to make in our pathway, is ATP, or adenosine triphosphate. This is, excuse me, this is our metabolic money. Through metabolism, we can, just like with regular money, we can earn it, we can bank it, we can save it, we can spend it, and we can exchange it. ATP acts as a temporary energy repository from one place to another. ATP can be made in one reaction, held in the bonds of the phosphates, and taken to another reaction where it can be spent. It is a three-part molecule. You have a nitrogen base, you have the which is adenine, you have a 5-carbon sugar, which is a ribose, and then you have a chain of phosphates. That chain of phosphates has 2 phosphates that are bonded together that give you high energy bonds, and the bonds, once those bonds are broken, energy is released and available to do work. So here we have our adenine, we have our ribose, and then we have our 1, 2, 3 phosphates. If you'll look here, between the first and the second phosphate we have a high energy bond, and between the second and the third phosphate we have another high energy bond. When those bonds are broken, energy is released, and now that energy is available to do whatever work is required for that cell. The high energy in ATP originates from the orientation of those phosphates. If I break the bond between the third and the second phosphate, then ATP is converted to ADP. Energy is released from that high energy bond and is available to do whatever needs to be done. I can also break the bond between the first and the second, and I convert ADP to AMP. All of these chemical reactions have to be replaced. I can't survive, or any cell can't survive, on adenosine monophosphate. So you're going to have reactions that are going to have to replace those phosphates, taking AMP back to ADP and ADP back to ATP, so we have the amount of energy required to go through metabolic processes. So if we start with ATP, and we need the energy available, the first thing that's going to happen is we're going to remove a phosphate through what is called dephosphorylation. Once that phosphate is broken, then energy is released from the bond of that phosphate, and we are left with ADP. Once that ADP is made, we need to convert it back to ATP. So we're going to go and we're going to take a phosphate, maybe from some other metabolic pathway. We're going to take energy that's produced from another metabolic pathway, and we're going to glue this phosphate onto ADP through a process called phosphorylation, and we will end up then with our ATP back. Sorry, I screwed that up a little bit, but hopefully you can follow that around and see how it works. ATP is the primary currency of the cell. It is constantly being broken and remade and broken and remade all the time. In heterotrophs, this energy comes from a number of catabolic pathways through aerobic respiration. There are 2 pathways that can produce ATP through the process of phosphorylation. The first one is substrate phosphorylation, where you have an enzyme breaking off a phosphate that comes from a substrate and attaching it directly to ADP. This would be substrate phosphorylation. Then you have oxidative phosphorylation, where you have a series of oxidation reduction reactions that will take the energy through that reaction and phosphorylate ADP to ATP through a series of oxidation reduction reactions. So that would be oxidative phosphorylation. Remember, if you have a substrate, you must have an enzyme. So if you're talking about substrate phosphorylation, those are going to be those ATPs are going to be formed when you have a series of substrate or enzyme catalyzed reactions. Oxidative phosphorylation is going to be when your series of reactions are oxidation reduction reactions. That is the easiest way to tell the difference between the two. Here's a picture. This picture is in your book. I believe it is on page 211. Essentially what happens is you have the phosphate removed from the molecule that is attached to. It's the other reactant within the chemical reaction is ADP. So once you have that catalyzed reaction, your now products are what is left of your original molecule and then ATP. Okay, I'd like you to take some time right now to maybe pause the PowerPoint, go over this information that we've talked about, and then write down any questions you have so that we can go over these and you'll understand what's going on, and we can make sure that you're not working on material that you don't quite understand. So take some time, write down any questions, and then we'll go from there. For those of you that are ready to go on, let's talk about the different pathways of metabolism. Metabolism uses enzymes to catalyze reactions. They're going to break down organic molecules like glucose into those precursor metabolites. These are catabolic reactions. These cells then are going to use these metabolites to build bigger, larger, more complex molecules, and this is anabolism. NAD, which your book calls reducing power, and energy are going to be needed to facilitate these anabolic parts of metabolism. Energy and NAD are going to be produced through pardon me, through these catabolic reactions. Anytime we get NAD and a reduced NAD and ATP, that reaction is a catabolic reaction, and that ATP can be banked then to use for any kind of anabolic reaction. Most of the time, we are going to use glucose as our form of initial energy. This is going to be the carbohydrate of choice of most organisms. Now we have metabolism for proteins, we have metabolism for lipids, but we're just going to talk about the metabolism of glucose. This is where we gain most of our energy, is through the breakdown of glucose. The most common pathway in the breakdown of glucose is called glycolysis. There are three major pathways in which we will break down glucose. The first one is aerobic respiration, which is the one we are going to spend most of our time discussing. This is a series of reactions that is going to convert glucose to CO2 and the end product of water, and it's going to allow the cell to recover significant amounts of energy that are held in as free energy in the form of hydrogen within this glucose. If you think about the chemical formula of glucose, it's C6H12O6. We have 12 hydrogens there that contain free energy that we are going to exploit through aerobic respiration. The second pathway that we can get some energy from is fermentation. When you have facultative or aerotolerant anaerobes, they can use the glycolysis portion of aerobic respiration to incompletely oxidize glucose. It will remove some of the energy, but not as much as you get from aerobic respiration. And then anaerobic respiration will generally go through glycolysis just like it does with aerobic respiration, but instead of using oxygen as a terminal electron acceptor at the end of the metabolic pathways, it's going to use some other final electron acceptor like sulfates or nitrates or carbonates rather than oxygen. Here's a great picture that talks about aerobic versus anaerobic fermentation. And so in aerobic respiration, you're going to see that you're going to go through something called glycolysis, then a reaction, the acetyl-CoA reaction, which is called the transition reaction, the Krebs cycle, and then electron transport with a terminal electron acceptor of oxygen. At the end of this, we will produce in bacteria 38 ATP. In anaerobic respiration, you're going to see we're going through the very same process, But instead of having oxygen as our terminal electron acceptor, we're going to have either sulfate, nitrate, or carbonate as our terminal acceptors. And ATP is going to vary based on your electron acceptor. You're going to either have 2 to 36 different ATP molecules made through anaerobic respiration. And then in fermentation, instead of going through the transition reaction and the Krebs cycle, you have glycolysis and then fermentation into lactic acid or ethanol. You can also produce other alcohols and acids from the fermentation process, but these are the two most common. The only ATP that you're going to produce is the ATP produced through glycolysis, and you end up then with a very limited amount of ATP. There is no terminal electron acceptor except for these organic molecules like lactic acid and ethanol. Those are the terminal electron acceptors in fermentation. I would take some time to really look over this picture. This diagram is on page 212 in your textbook. It's a very good picture to understand and use as we follow through the pathways of metabolism. In aerobic respiration, we're going to have a series of these enzyme catalyzed reactions. We're going to transfer electrons from molecules like glucose to oxygen, which is our terminal electron acceptor. This is the principal energy producing mechanism for chemoheterotrophs, those aerobic chemoheterotrophs. It provides both ATP, it provides those metabolic precursors, and it is going to be the one pathway or set of pathways that will break down glucose the most efficiently. If we're going to start with the whole process of metabolism, we're going to be breaking down glucose, and the first process that we're going to use is a pathway called glycolysis. Glycolysis will take glucose, and in order to spend, basically it is going to convert glucose through a series of steps to produce an end product of pyruvic acid. Once we do this, we then can move this on to another pathway. With glycolysis, we're going to take our glucose and we are going to spend 2 molecules of ATP to break it down to 2 intermediate molecules of glyceraldehyde. Now, if you look in your book, you're going to see that there are a number of different steps that I have left out. I have even actually redrawn this whole process so that you can see what's going on step by step in glycolysis. In step 1, ATP is going to be expended to add a phosphate group to our glucose. Then we're going to expend a second ATP to add a second phosphate to our glucose. The energy that was released through the dephosphorylation of ATP has split this 6-carbon molecule into 2 molecules of glyceraldehyde. Now, I started with 6 carbons and I have to stay with 6 carbons. I have to be able to keep track of all of these. So I may have 2 molecules, but I still have my 6 carbons available. So we have split glucose into glyceraldehyde. Now remember, my end product for glycolysis is pyruvic acid. So I'm going to take my glyceraldehyde now and I am going to go through an oxidation reduction reaction where my glyceraldehyde will give away a hydrogen. When it gives away that hydrogen, it's taking its electrons with it. So if something is losing an electron, it is being oxidized and something else must be reduced. So our glyceraldehyde is going to be oxidized. NAD, our coenzyme NAD, is going to be reduced to NADH. Wherever the hydrogen ends up, that's the atom or molecule that has been reduced. So if NADH is our reduced molecule, then you know that glyceraldehyde had to be oxidized. From this point, we're going to have a series of enzyme catalyzed reactions that are going to allow for the phosphorylation of ADP to ATP. So we are going to phosphorylate these, and we know that they are substrate phosphorylated because these are a series of enzyme reactions. Not whatever we do to one side, we have to do to the other. So I have now 2 molecules of ATP. I'm going to do this again. I'm going to phosphorylate ADP to ATP through another series of reactions, and I will end up with my final oxidized product of pyruvic acid. This is the end, essentially, of glycolysis. So I have produced 2 molecules of NADH, which is a reduced coenzyme. These NADH molecules are going to be released, and they are going to be taken someplace else where we will convert them to ATP. Remember that the hydrogens are free energy. That free energy now is going to be put in the form of ATP, and we're going to do that using this coenzyme. So we're shuttling these electrons from glycolysis to another set of reactions. Pyruvic acid is our end product here. So we are ready to go on to the next step in metabolism. Pyruvic acid, basically from glycolysis, serves a very important position in a number of pathways. We can go aerobic, we can go anaerobic, and now we can even go fermentation. So different organisms are going to handle it different ways. When we're talking about aerobic organisms and anaerobic organisms, these are going to enter into the Krebs cycle. Okay, from this point, we are going to either choose to go aerobic or anaerobic, depending on your organism. If you are a facultative organism, from this point, you're going to go into fermentation, where you're going to either produce lactic acid or ethanol. In pyruvic acid, we made 2 molecules of NADH, which is our reduced coenzyme that is going to shuttle these high energy electrons to someplace else. We made 4 ATP in glycolysis. There is a net production of 2 ATP because we have to pay the 2 back that we used at the beginning of the cycle. If you remember, we went back to the beginning of that cycle. We took our glucose and we had to add 2 molecules of ATP to split that glucose in half. ATP is then going to be produced through substrate phosphorylation of ADP. So we took ADP, we added a phosphate through our enzyme reactions, and now we have 2 net production of ATP. So once we have glycolysis, we now need to be able to choose which way we're going to go. So we can go through aerobic respiration. We can go through some anabolic pathways where we can basically continue to break down sugars. We can go and start breaking down fat molecules as well as protein. We can take the other way and we can go through fermentation. So it's at this point that we have to decide which pathway we're going to take. We are going to decide to go through aerobic respiration. Once we have our pyruvic acid, there is a lot of energy still remaining in the bonds of that pyruvic acid, and it's going to be transferred to the Krebs cycle. Pyruvic acid is going to be oxidized at the beginning of this cycle, and it's going to reduce NAD to NADH. That NADH is going to take that hydrogen someplace else where we can convert it or produce ATP. If we are bacteria, the Krebs cycle takes place in the cytoplasm. If we are a eukaryotic organism, it's going to take place in the matrix of the mitochondria. Here we are going to produce a number, considerably more reduced coenzymes. We're going to produce 8 NADH and 2 molecules of FADH. This is a new coenzyme, and I'll explain a little bit about this as we continue on with the Krebs cycle. And we're going to produce 2 molecules of ATP for every glucose molecule that we started out with. ATP is going to be produced through the substrate phosphorylation of ADP to ATP. Now I know this is sounding like a bunch of gobbledygook, but let's look at a diagram and see how this works. We start out with pyruvic acid. Pyruvic acid is a large molecule, relatively speaking, and in the Krebs cycle, it only has room for a certain size of a molecule. So we have a reaction here called the transition reaction. And this transition reaction is going to give us an end product of acetyl-CoA. And what we have to do here is we are going to now oxidize pyruvic acid, meaning we're going to take away a hydrogen, and we are going to reduce NAD, which is our coenzyme, to NADH. When we do this, we are also going to add another molecule called coenzyme A. Coenzyme A is going to initiate the chemical reaction that's going to take place to produce acetyl-CoA. At the same time, we're going to release one of our carbons as carbon dioxide, and we end up now with a 2-carbon molecule of acetyl-CoA. This transition step is going to give us the starting molecule of the Krebs cycle, or the citric acid cycle. Okay? Once we have our acetyl-CoA, we are ready to start this cycle. And for every pyruvic acid, you are going to have this cycle go around 1 time. So we are going to look and see, we've got 3 pyruvic acid, or we've got 3 carbons in our pyruvic acid. We've lost 1 to carbon dioxide. We have 2 of our original carbons left, and we're going to need to be able to metabolize and release those at the same time. So we are going to take our acetyl-CoA, and this enzyme, or this molecule, is going to bind with the last 4-carbon molecule of the previous cycle. This is a cyclic reaction, so as it goes around, the last molecule from the previous cycle now is going to bind with our acetyl-CoA, and that's going to give us a new 6-carbon molecule to begin our Krebs cycle. 2 of these are from our original pyruvic acid. 4 of them are a molecule that will begin to stay within the Krebs cycle. So from this 6-carbon molecule, we're going to go through an oxidation reduction reaction, and we are going to oxidize this 6-carbon molecule, and we're going to reduce NAD to NADH, and we are also going to release another carbon dioxide. So now we have released 2 of our carbons from our initial pyruvic acid. We are now down to a 5-carbon molecule. This 5-carbon molecule, we're going to go through the same process of oxidation and reduction. We're going to oxidize the 5-carbon molecule. We're going to reduce NAD to NADH, and we're going to release our last carbon dioxide. And we have now metabolized all of the carbons out of our pyruvic acid. What's left are our high energy hydrogens. So we've got a lot of work we still have left to do here. At this point, we have a 4-carbon molecule, and this 4-carbon molecule now can act as a substrate, or a substrate for the phosphorylation of ADP to ATP. When we do that, we have this reaction that gives us ATP and a new 4-carbon molecule. As these chemical reactions have been taking place, these reactions are going at a lower and lower and lower energy as they go. We are now down to a 4-carbon molecule that is at a very low energy. NAD is a coenzyme that can only carry specific hydrogens that have a specific energy level. So what we're going to have to do is bring in a new coenzyme that can take on these lower energy hydrogens, and that is going to be the coenzyme FAD. FAD, we're going to start at this new 4-carbon molecule. We're going to oxidize this 4-carbon molecule. We are going to reduce FAD to FADH, and we have a new 4-carbon molecule. This 4-carbon molecule is extremely low on energy. There is no coenzyme that will take on these low energy hydrogens. So we have to give it a source of higher energy hydrogens, and the way we're going to do that is to add a molecule of water. When we do that, that brings these gives us a new 4-carbon molecule. It brings these 4-carbon molecules up to a higher energy level. Now we can go through a higher oxidation reduction reaction, and we're going to take our last 4-carbon molecule, we're going to oxidize it and reduce NAD to NADH again, and we are back at the beginning of our cycle with the last 4 carbon molecule, and it'll be ready to go in and bind with acetyl-CoA and start the process all over again. Now I know a lot of you are going, I don't get this. I'm going to take some time on Thursday to go over this with you step by step by step so that you understand what you are looking at and exactly what is going on. Hopefully these pictures are allowing you to go through and you can see them. I've also got some animations that I will put out there. I probably will have to send them to you on email so that you can see how all of these reactions work. Okay? Let's go ahead and see what happens after we get all of this NADH. We have all of this NADH, we need to know what to do with it. So let's go from there. The last portion of aerobic respiration is something called the electron transport chain. We have had a few molecules of ATP, and remember the purpose of metabolism is to produce ATP. Well, at this point we've only made 2 in glycolysis and 1 for each of the Krebs cycles, so we've only made 4 ATP. This is a lot of work for 4 ATP. So we now are going to take all of this NADH and all of this FADH and we're going to put it to work. This electron transport chain, your book actually calls it the processing mill of electrons and hydrogen atoms. And I'm sorry, there is an error here on your page. Basically, we are going to generate, I'm sorry, no there's not. We are going to generate 34 ATP in bacteria, 32 ATP in eukaryotic cells. Combining that with the ATP that we've already made in glycolysis in the Krebs cycle, we will make our 38 and 36 molecules of ATP. Basically what the electron transport chain is a chain of oxidation reduction reactions as you have these redox carriers receive and give away electrons from NADH and FADH. Essentially what is happening is once NADH and FADH are made, they are leaving the pathway that they were formed in, moving to the electron transport chain where they will be reoxidized by giving away their high energy electrons, and those electrons then will be carried down a transport chain where they will end up with oxygen to produce water. And in aerobic respiration, the terminal electron acceptor is oxygen, and you end up then with water. Unfortunately, I don't have a spiffy little diagram to help you go through one at a time. But essentially, if we look here, we see here this first one is the mitochondria of the eukaryotic cell, and here we have the plasma membrane and cell wall of the prokaryotic cell, but things work generally the same. What's going to happen is we're going to take our hydrogens from our NADH and we are going to strip them of their electrons. The protons are going to be shoved to the outside of the membrane, and the electrons are going to be passed down this set of carriers until it gets to an enzyme called ATP synthase. As we have all of these hydrogens accumulating on the outside of the membrane, and if I come down here in the bacterial one, it looks a little more obvious, but you have these hydrogens now that have accumulated here, and this produces something called the proton motive force. That proton motive force now is going to be sent through this ATP synthase, and that ATP synthase is going to allow for the oxidative phosphorylation of ADP to ATP. The electrons that were stripped from the hydrogens now have been added back to them. If we come back up here, we can see in here you have these electrons that are going to be added back to this hydrogen, and that hydrogen is going to add itself to bond itself to oxygen, and we end up with water. Okay, so essentially all we're doing is we're passing electrons off as we go. For every NADH that gets reoxidized through the electron transport chain, you're going to produce 3 molecules of ATP. for every FADH that goes through the electron transport chain, you're going to make 2 ATP. You'll see here at this top picture, NADH goes up through an enzyme called NADH dehydrogenase. Well, this molecule, or NAD, has, remember, those higher energy electrons, so it's going to go in at a higher energy level on the electron transport chain. FAD is going to go through a carrier on a membrane called FMN, flavine mononucleotide. And essentially what happens here is the same thing to NAD. But this is a lower energy molecule than the NAD dehydrogenase, and that is where FAD goes in. So you have less energy coming through FAD than you do NAD. So for every NADH that is reoxidized, you get 3 ATP. For every FADH that is reoxidized, you get 2 ATP. And if we go through all of the math, you will see that we will end up with 34 and 32 ATPs respectively, but depending on whether you're eukaryotic or prokaryotic. The summary of the electron transport chain is essentially energy is produced through the series of oxidation reduction reactions. Those electrons move through those membrane proteins until we get to the end, and they are accepted by the terminal electron acceptor, in this case, which is oxygen, and we're going to produce water. 3 molecules of ATP are going to be generated for every NADH that's reoxidized, and we make NADH in glycolysis and the Krebs cycle. 2 molecules of ATP are going to be generated for every FADH that is reoxidized, and FADH is produced in the Krebs cycle only. 34 ATP are produced in the electron transport chain through the reoxidation of reduced NADH and FADH. So if we look at a summary of phosphorylation in aerobic respiration, what we have is produced from 1 molecule of glucose, we have in glycolysis 2 molecules of NADH. We have a net gain of 2 molecules of ATP through substrate phosphorylation. And in the transition reaction and Krebs cycle, we have 8 molecules of NADH. We have 2 molecules of FADH and 2 molecules of ATP through substrate phosphorylation. And in the electron transport chain, we have 34 molecules of ATP through oxidative phosphorylation. So if we take these 34 plus the 2 from the Krebs cycle, that gives us 36, plus the 2 from glycolysis, that gives us 38 ATP. Here is a table. This is kind of a weighty table, but it does give you an idea of where things happen, what the reactants are, what the intermediate products are, and the end products of the whole process. So I would probably take some time to go over this. I will go over it with you in class. I will probably even give you a bit of an abbreviated table that will help you a little bit in understanding what's being made and where it's being made and what exactly is going on in this whole process. Why the difference between 32 and 34 ATP for eukaryotic versus prokaryotic? In the eukaryotic cell, the 2 NADH from glycolysis have to move into the mitochondria to get to the electron transport chain. Glycolysis takes place in the cytoplasm. These do not have as much energy as the other NADH, so only 2 ATP are produced from the 2 molecules of NADH from glycolysis. So if we look here, we have 10 NADH in prokaryotic cells times 3 ATP. That gives us 30 ATP. We have 2 FADH times 2 ATP, and that gives us 4 ATP in prokaryotic cells. So we produce 34 ATP through the electron transport chain in prokaryotic cells. In the eukaryotic cell, we have our 2 cells from glycolysis, which are only going to give us 2 ATP. So we have 4 ATP there. we have 8 from the from the Krebs cycle. 8 times 3 is 24, and then our 2 FADH, again, we get 2 ATP from there, and that gives us 4 ATP. So 24 times 4 is 28, times 4 is 32. So we get that whole difference between the 32 and the 34 is based on the movement of those original NADH from glycolysis. They don't have as much energy once they get into the mitochondria because they have to expend some of that energy coming across the plasma, the membrane of the mitochondria. So let's look at a total summary of aerobic respiration. There's a total possible yield of 40 ATP, 4 from glycolysis, 2 from the Krebs cycle, and 34 from the electron transport chain. But because we used 2 in glycolysis, we had to pay 2 back, so that gives us a total net yield of 38. We have 6 carbon dioxide molecules that are going to be generated during the Krebs cycle. Remember, for each pyruvic acid, we released 3 carbon dioxides. We had 2 pyruvic acids, so therefore we have 6 carbon dioxides. 6 oxygen molecules were consumed during the electron transport chain. 6 molecules of H2O, or 6 molecules of water, were produced in the electron transport chain. 2 in glycolysis, but 2 were used in the Krebs cycle for a net of 6 molecules of water. So if we look at the total process, we have 1 molecule of glucose plus 6 oxygen will yield 6 carbon dioxide and 6 water and 38 ATP as a prokaryotic cell. As a eukaryotic cell, it's 1 glucose plus 6 oxygen will yield 6 carbon dioxide plus 6 water and 36 ATP. Anaerobic respiration functions just like aerobic respiration, except for the fact that that oxygen is going to be not going to be the terminal electron acceptor. You're going to use some sort of oxygen containing ion like sulfate or nitrate or carbonate, not free oxygen. The nitrate-nitrite reduction systems for anaerobic bacteria are the best ones that have been known and understood. And these systems are going to use a set of enzymes called nitrate reductase enzymes. This process actually leads to something called denitrification, where you have enzymes that can reduce nitrate to nitric oxide and nitrous oxide and nitrogen gas. And this is important in recycling nitrogen back into the atmosphere. So when we talk about bacteria in plants and animals and things like that that are nitrogen fixers, these are the ones that are actually bringing the nitrogen back into the atmosphere that keeps that nitrogen level consistent. And so a lot of the anaerobic organisms have a big part in the whole denitrification and the nitrogen cycle of nitrogen fixation. If we're talking about fermentation, this is basically going to be the incomplete oxidation of glucose or whatever carbohydrate, whether it's lactose or sucrose or maltose or whatever. But this is all going to take place in the absence of oxygen. It's going to use some sort of organic compound as a terminal electron acceptor instead of oxygen. A good amount of the time it's going to be lactic acid or ethanol, which are going to be the most common. You have a very small amount of ATP produced here, but you can have a lot of bacteria that can grow just as fast utilizing fermentation as they do aerobic respiration because it causes an increase in the rate of glycolysis. Remember, we talked about those pacemaker enzymes, and that environment is going to have a big effect on the rate of enzymatic reaction. The absence of oxygen is essentially going to increase the rate of glycolysis so that it can keep up with the energy demand of the organism. This basically is going to permit the independence from molecular oxygen by a number of bacteria. So this is where some of these facultative organisms can grow the very best. We have a number of products of fermentation. Alcoholic beverages is probably the most important. Organic acids, which can lead to the production of dairy products. We have vitamins, antibiotics, even some of our artificial hormones. There are two general categories of fermentation. You have alcoholic fermentation, and then you have acidic fermentation. In alcoholic fermentation, basically you have yeast or some sort of bacteria that can use the metabolic pathway to convert pyruvic acid into ethanol. And the products that you have then are ethanol and carbon dioxide. So if we start with glycolysis and we end up with our pyruvic acid. So it's gone through the whole process of glycolysis. We have our two pyruvic acid molecules. The first thing that we're going to have is something called decarboxylation, where we're going to remove carbon dioxide, and we end up with an intermediate molecule of acetaldehyde. That acetaldehyde now is going to act as an electron acceptor. So instead of being oxidized, this molecule is going to be reduced. We're going to add a molecule of NADH to each of the acetaldehydes, and that NADH is going to be oxidized by giving up its hydrogen to NAD+, and our final reduced product is ethanol. Much easier than aerobic respiration. If we're talking about lactic acid fermentation, essentially pyruvate is going to be reduced straight to lactic acid. You have heterolactic fermentation when glucose is fermented to a mixture of acids of lactic acid, acetic acid, and carbon dioxide. We can also have mixed acid fermentation where you have a combination of a number of acids that are produced, acetic, lactic, succinic, and formic acids. This is going to lower the pH of a particular growth medium to a very relatively acid pH. So if we start with glycolysis again, we end up with our pyruvic acids. This pyruvic acid now is going to act as the electron acceptor. This pyruvic acid is going to be reduced. NADH is going to be reduced to NAD, and we have an end reduced product of lactic acid. This is the end of metabolism. I know we kind of raced through this, but I would encourage you to go through this again step by step, pausing, taking down notes, writing any sort of questions that you might have, and then we will go through these step by step in class the next time we see each other. This is a relatively easy material to understand if you can look at it and go one step at a time, and we can ask questions as we go. So we're going to do that on Thursday when I see you on this next Thursday. So take time, watch it, read it, listen to it, stop, write your questions, and we'll go through them all next time we see each other.